Thank you.
Okay, welcome everybody who's kind of in the process of joining. So this is Imaging ONEWORLD again, which I think hopefully a lot of people in the imaging community are really aware of, and many of you have joined. You may have noticed I haven't actually been joining so many of the events because a new job has actually been interfered with my schedule. So but I can join most of the kind of meetings to come for at least the first half hour. So that's why today you have Nick Barry providing you with the quiz, and hopefully let's see how well it goes, and we hope it's enjoyable after the talk. But first of all, to introduce Stephan Uphoff. He's joining us from Oxford, where he's a group leader in biochemistry. He has studied physics in Göttingen and specialized in single molecule biophysics. When he went to Oxford for his MSc and PhD. And he also moved to Harvard for postdoctoral research in the field of systems biology, but did return to Oxford with the biochemistry department, the Wellcome Trust, as a Wellcome Trust fellow. And he really had established it in this independent group in 2017. He has won numerous prizes for his work, the Colworth Medal of the Biochemical Society and the Lister Institute Research Prize and the Wellcome Wade Prize. So his multidisciplinary research group is doing really wonderful new imaging techniques and applying them at the moment, and what we will see today, to bacteria. And he's imaging how bacteria sense and repair DNA damage at the single molecule level. And please, Stephan, tell us about the wonderful research and show us some of those beautiful images.
Thank you very much, Stephanie. That's a really kind introduction. It's a really great pleasure to be here online today and reach the community of microscopy enthusiasts. Thanks also to Nick and Katie for the invitation. So I'm going to talk about our application of single molecule and single cell live cell imaging technique to image how bacteria sense and repair DNA damage. And so just by way of introduction, let's get started with a very sort of broad level overview why DNA repair matters. So of course, there's this important balance between genetic stability and plasticity that's right at the core of life and evolution. So genetic stability is crucial. Every time a cell divides, it needs to accurately copy and then segregate all of its genetic material. And so in a multicellular organism, from one fertilized cell to a full human, this goes from one cell to 10 to the 13 cells. And crucially, the genetic sequence needs to be maintained throughout all of those cell divisions, and then passed on from parent to children. And so this obviously requires error correction mechanisms to maintain the DNA sequence information. But on the other hand, for life of species on the planet, it's also been very, very important to have genetic plasticity. And that's the ability for DNA sequence to change over time. Species can mutate and they adapt to new environments. And this balance between, on the one hand, genetic stability and on the other hand, genetic plasticity is maintained by the processes of DNA repair and mutagenesis. So you'd imagine that normally cells would benefit from maintaining genetic stability via DNA repair mechanisms. But maybe when conditions change and an organism might be poorly adapted to its current environment, then it might be beneficial actually for mutations to arise, which could increase the chance of adaptive mutation coming up, which may benefit the organism to live in that new and changed environment. And so it's becoming evident that the balance between DNA repair and mutagenesis is not fixed in time, but that it can be actually modulated according to the environmental conditions that an organism finds itself in. And so where do mutations come from? So fundamentally, of course, we know that mutations come every time the DNA sequence is copied by DNA polymerases. There's a chance that the DNA polymerase misincorporates a wrong nucleotide. So putting a G opposite a T or an A opposite a G, which would be initially a DNA mismatch. And this DNA mismatch, through another round of DNA replication, can become a fixed mutation. Now, the frequency of these DNA replication errors is very, very small. So DNA polymerases have evolved to be very, very accurate. But on the other hand, the DNA, the parental DNA that is being copied before cell division is not always perfectly intact. There are various different types of DNA damage. And these DNA damages can perturb the fidelity of DNA replication. They can increase the chance that DNA polymerase misincorporate the wrong nucleotides. And so cells have evolved DNA repair mechanisms that remove these DNA damages such that they do not interfere with the process of DNA replication. But on the other hand, DNA damage is being sensed by organisms and induces DNA repair mechanisms. So from these few errors, you can already imagine that there's quite a complex interplay between the amount of DNA damage, the amount of DNA repair that's going on, and ultimately what fraction of DNA damage sites could be converted into mutations. So it's quite a complex interplay between the sensing of DNA damage, the amount of DNA repair, and finally mutations being generated. Furthermore, not all DNA repair mechanisms are perfectly free, perfectly accurate. They're actually error-prone DNA repair pathways, which help cells to complete DNA replication, but these mechanisms are themselves error-prone, so they actively facilitate the formation of mutations. And they benefit the completion of DNA replication at the cost of accuracy. And now, this is particularly interesting in the context of treatments of cancers and treatments of bacterial infections with drugs that try to kill cancer cells or bacterial pathogens by creating DNA damage, because DNA damage is toxic, it interferes with DNA replication and can lead to the death of the cells. And so this is the benefit of why many chemotherapy drugs are used to treat tumors generate actually DNA damage. Many different types of antibiotics also directly cause DNA damage or generate DNA damage as potentially a side effect of the treatment. But an unwanted side effect of that DNA damage is that it not only kills cells, but it also actively promotes the formation of mutations. And these mutations thereby accelerate the evolution of resistance to these treatments. So we are in my lab particularly interested in bacteria. And bacteria in their environment are frequently exposed to different types of stress conditions. That could be starvation, changes in pH or temperature, exposure to radiation, infection with phages. These are the viruses that infect bacteria, different types of reactive chemicals like reactive oxygen species, also competition between different bacteria, of course the host immune responses that fight bacterial pathogens, treatment with antibiotics, and also aging in bacteria. All of these different types of stresses elicit a stress signal inside the bacteria. The bacteria then launch a stress response, which tries to switch off the stress signal by either repairing the cellular damage that's caused by the stress, shutting down stress-sensitive processes, eliminating harmful agents from the cell, combating aggressors, whether they have phages or other bacteria, or the immune cells, and ultimately by mutating and adapting to become resistant to the type of stress. Now, this ability to launch stress responses allows cells that have a particular genotype to switch from a stress-sensitive state to a more stress-tolerant state. However, when the stress is really severe, then such a stress-tolerant state might not be sufficient for bacteria to survive. Instead, they rely on mutation to a new genotype to become stress-resistant. And it's interesting to think about the maintenance of genome stability or plasticity as itself a phenotype that the cells express. So during normal growth conditions where the cells are perfectly adapted to their environment, cells might favor genome stability, maintenance of the DNA sequence. Whereas when conditions are poor and an organism is maladapted, then it might benefit from switching to a mutative phenotype, and such a mutative phenotype then increases the chance that a new mutation to a beneficial genotype that leads to stress resistance arises. And now we're interested in this interplay, and because it's quite complex and many different parameters play into this, we want to see this at the level of living cells and directly resolving the molecular processes inside the living cells. So many years ago, we started development of single molecule imaging technique that allow us to observe DNA repair reactions directly using single particle tracking. So this relies on some custom-built single molecule fluorescence microscope using total internal reflection illumination. And we tested this technique initially by looking at a DNA polymerase that binds to gaps in the DNA, which are generated when a DNA lesion is excised by a particular DNA repair pathway, which recognizes these lesions, cuts that out, leaves a gap in the DNA, and this gap in the DNA can then be filled in by a DNA polymerase. And so in our early work, we fused the DNA polymerase Pol1 in E. coli to a photoactivatable M-cherry fluorescent protein. And this allows us to visualize individual fluorescent molecules one at a time, because the photoactivatable M-cherry is usually in a dark state, but can be activated to a fluorescent state one molecule at a time. So here in the top, I'm going to start the movie, are just two E. coli cells, where at some point in the movie, a single Pol1 PA M-cherry molecule becomes fluorescent. So there's a bit of background noise to start with, and now a single molecule has become fluorescent. You can see it's diffusing around in the living cell. We can follow its motion. In each frame of this movie, we find the position of the molecule, and then we use an algorithm that tracks the positions from frame to frame. And then you end up with something like the image on the right, where you see these sort of blue spaghetti lines that go all the way through these individual E. coli cells. These are the tracks of these individual molecules that become photoactivated at different times throughout a movie. Now, when we treat the cells with a DNA damaging agent called MMS, which is short for methylmethane sulfonate, it's a DNA damaging agent that causes methylation of DNA bases. These methylated DNA bases are cut out and leaving substrates for the DNA polymerase to bind to. What we then see is that individual photoactivated molecules now become immobilized. Now we see a mixture of diffusing molecules, which are plotted in blue, and then we see these immobilized molecules, which are not moving much during the time that they are photoactivated for. And these are plotted in red. So you can see that after DNA damaging treatment, we see a lot more of these immobilized single Pol1 molecules. And this is a very general technique from which you can obtain quite a lot of different information. You can learn about the diffusion constants of the molecules. You can ask where the DNA damaged sites are that the proteins are binding to, how many there are per cell, whether the differences between cell to cell in the amount or distribution of DNA repair sites, etc. So with this technique over the years, we've looked at a bunch of different types of DNA transaction, from DNA replication to mismatch repair, base excision repair, processes involved in transcription, and also chromosome organization and segregation. Here I'll just highlight one such study where we looked at the initiation of nucleotide excision repair. And if you look at the Wikipedia article or other earlier literature, you read that nucleotide excision repair in E. coli is initiated by a complex of two proteins, UVRA and UVRB, which are believed to scan the genome looking for sites of DNA damage to bind to. And when that has happened, then a third protein gets recruited, UVRC, and this protein that makes two cuts in the DNA strand on either side of the lesion, which then allows that strand to be removed, leaving a gap for the DNA polymerase to fill in and then the ligase to see it. Now, in collaboration with others, we looked at the distribution of the DNA content in the cell using a dye that binds to the DNA. It's an intercalating dye. So we can see where the DNA is in the cell. And then in the same cells, we used photoactivated single molecule tracking when using a fusion of UVRA to the PA M-cherry fluorophore, and did the same tracking experiment that I showed on the previous slide. So we can see immobilized molecules here shown in red in this case, and diffusing molecules in sort of blue to green, according with the tracks colored according to the diffusion coefficient. Now, in a separate experiment, we looked at the distribution of DNA and the diffusion coefficients of UVRB. And what we see here is quite a different distribution. UVRB molecules are diffusing much faster, so you can see them mostly in green color here. And these tracks of UVRB seem to fill the entire volume of the cell, whereas the tracks of UVRA seem to be localized, mostly where the DNA is. And in fact, when we look at the distribution of diffusion coefficients for UVRA and UVRB, we can see that they're very, very different. UVRA has very slow-moving molecules, whereas UVRB has a more faster diffusion inside cells. And this, together with several different control experiments and other perturbations, genetic perturbations of the cells especially, we developed a model that actually finds UVRA searching for DNA lesions on its own, in close contact with the DNA, staying where the nucleoid is, searching along the DNA for lesions, whereas UVRB diffuses everywhere in the cytoplasm, that is both where the nucleoid is and where it isn't. It's diffusing around, and then it only binds to lesions after UVRA has already located and recognized the lesion. This is something that would have been difficult to discover like this from in vitro experiments, because in vitro experiments can find conditions where UVI and UVRB do form a complex and bind to lesions together. However, it's unclear whether these are the conditions that are found inside cells. So only by live cell imaging, using fusions of the native enzymes, can we have conditions where the expression levels of the enzymes are unperturbed, and we can directly compare the mobility and localizations of these proteins in the native cellular context. So now, with the single molecule tracking experiments, you can start to see directly individual molecules as they're searching for DNA lesions, like this DNA polymerase that's diffusing around in these light blue tracks, and then finds a lesion, it binds there for some time, then unbinds and searches for the next lesion. So looking at tracks like this, of course, you wonder, can we learn something about this, about how the DNA polymerase identifies lesions inside cells? And it's been long suggested that many different types of DNA binding proteins, including DNA repair enzymes, search for their lesions or target sites by combining a 3D diffusion in the cytoplasm with a non-specific binding to DNA, and then sliding in 1D Brownian motion search along the DNA, then unbinding again, binding somewhere else randomly, diffusing along the 1D sequence until a target site is reached. But it's been very difficult to figure out whether this motion actually occurs inside living cells, and what's the relative balance between how much time is spent freely diffusing and how much time is spent non-specifically binding and sliding along the DNA. So to address this question, we looked at the diffusion coefficients of the DNA polymerase 1 initially, and here's the distribution of the diffusion coefficients, and we fit this with a model for two different populations, the immobile population of DNA polymerase bound at target sites, and then a distribution of mobile molecules that diffuses around in the cell. We also look at the distribution of the tracks inside the cell, and you can already see that these tracks seem to be sort of organized in two lobes. These lobes very closely recapitulate where the DNA is inside E. coli cells. As the E. coli cells are replicating their DNA, they're segregating the genetic material, and they're going to divide in the middle of the cell after the two chromosome copies have been segregated to each half. So here we can see that the DNA polymerase molecules seem to stay in close contact with where the DNA is supposed to be inside the cell. Now, to test specifically the influence of the chromosome, the DNA, on the diffusive motion that we see for the DNA polymerase, we used a technique to generate chromosome-free E. coli cells. So for this, we genetically engineered the E. coli cells to have a restriction endonuclease, which generates a DNA double-stranded break on the chromosome. And then we used a strain of E. coli that is unable to repair such DNA breaks. What then happens is that the DNA break is generated, it cannot be repaired, and nucleases in the cell then digest the entire chromosomal DNA. So in about 60 to 90 minutes, cells that are initially intact, like on the left here, you can see the DNA stained with DAPI dye in blue, and the membrane stain just to show where the cells are. These cells are converted to chromosome-free cells, where the majority of the cells has completely lost the DAPI stain that shows the DNA. So these are chromosome-free cells. And now we perform the single molecule tracking experiments in these chromosome-free cells and compare them to the intact cells that have chromosomes. And so here in these chromosome-free cells, you can see that the DNA polymerase one diffuses about 2 times as fast, and also the immobilized population of molecules shown in red has been lost. And you can see that the distribution of the tracks now fills almost the entire volume of the cytoplasm. So from this by eye, you can start to guess that this DNA interaction has been lost. The diffusion coefficient has increased on average. The interaction, the spatial distribution of the tracks has been lost. So this indicates that the binding of DNA during the target search plays actually a big role. And in fact, by comparing the relative diffusion coefficients in the chromosome-free population to the intact cells, which is about a 2-fold difference, we can actually calculate what fraction of time the DNA polymerase is spending freely diffusing or non-specifically binding to DNA or specifically binding at target sites. And we find that for the DNA polymerase, about 60% of the time is spent non-specifically binding to DNA, which is the majority of time. Of course, the question was, this slowdown of diffusion that we see in intact cells, or conversely, the speed up of diffusion that we see in cells that do not contain a chromosome, is that due to genuine DNA binding, or is that due to a steric entrapment of the protein in the dense meshwork of DNA inside the cell? In other words, is this something specific to DNA binding proteins, or is that something that basically every protein in the cell would speed up once the chromosome has been degraded? So to address that question, we looked at the LAC repressor. The LAC repressor has long been a favorite model protein to look at target search processes because it searches for its particular promoter sequence on the chromosome. And so here we looked at the diffusion of the LAC repressor in intact cells on the left, and then in chromosome-free cells on the right. Again, you can see this big speed up in the diffusion of the LAC repressor in chromosome-free cells. And now to ask if this is due to DNA binding or steric entrapment in the intact cells, we used a mutant of LAC-I that lacks the DNA binding domain. So when the DNA binding domain is lost, then even in intact cells, we see much faster diffusion. And then importantly, when we look at this LAC-I DNA binding mutant in chromosome-free cells, we see no further increase in diffusion. So the diffusion of the DNA binding mutant is the same in chromosome-free cells or in intact cells, which shows that the steric entrapment does not play a role in the diffusion properties of this protein, but it is indeed the specific DNA binding that slows down the diffusion in intact cells. We then looked at a bunch of other DNA unbinding proteins to see how conserved and general this effect is. So we looked at the RNA polymerase. Here's the distribution of diffusion coefficients and the spatial distribution of the tracks in intact cells and then in chromosome-free cells. Again, here in this case, you see about a 3- to 4-fold increase in diffusion, and the tracks fit the entire cell. MUC-B is a DNA organizing protein that also speeds up a lot when the chromosome is lost. And here's the DNA ligase, which acts in DNA replication and repair. And again, you see an increase in the diffusion and the tracks filling the entire cytoplasm. And so from the ratio between the diffusion coefficients in intact cells compared to the diffusion coefficients in the chromosome-free cells, we can estimate the fraction of time that each of these proteins is spending non-specifically bound to DNA. We did this for many different types of proteins, and we find for all of the proteins, they spend the majority of time non-specifically bound to DNA. So this is above 50% for all these different proteins, which are involved in DNA repair, chromosome organization, replication, transcription, etc. For some proteins, this even reaches 90, even approaching 100% of time bound to DNA. And then by looking at the copy numbers of these proteins, so the number of these proteins per cell, and the footprint that they have on DNA, then we can estimate what amount of the chromosome is actually covered by these proteins. And we find that these proteins occupy 30% of the E. coli chromosome at any time, which is a really surprisingly large number. So it paints kind of a different picture of the target search of proteins for their target size. So we get the idea that proteins stay in very close contact with the DNA. They often bind transiently to DNA, unbind occasionally, but then rapidly rebind. So they're kind of performing a very local search, where as they are binding to DNA and potentially sliding in 1D, they might often return to the same site multiple times. But there will also often be collisions with other non-specifically bound proteins on the DNA. And overall, the chromosome is very much covered with non-specifically bound proteins. And that must also mean that often the target sites that proteins are searching for will also be bound and occupied by other non-specifically bound proteins. So this might create the impression that the search for DNA lesions of DNA repair proteins could be rather hopeless. How can we ensure chromosome stability and accurate removal of DNA damage in such a case? Well, you could just think, let's just overexpress these DNA repair proteins. The best strategy for cells might be to have very, very high numbers of these DNA repair proteins to make sure that lesions are not lost. But of course, that has energetic costs and also creates problem for all the other DNA transactions, because overexpressing one protein and then further crowds the chromosome and might perturb the action of all the other proteins. So instead, bacteria have opted for a different strategy in many cases, where they usually have very low expression of DNA repair proteins. But once they sense the presence of DNA damage, then they start to produce more proteins when needed. But of course, that comes at a risk. It comes at the risk that there might be a lot of DNA damage and the cells haven't rapidly sensed it or have not yet managed to induce sufficient numbers of DNA repair enzymes. So the cells don't really want to be caught in that state where they have lots of DNA damage but haven't yet produced enough repair enzymes to remove that damage. And so to look at the fidelity of this process and the links between the ability to sense and regulate DNA damage responses of cells, we looked at a model DNA damage response, one of the first DNA damage responses that was described and discovered back in the 70s. And this is the adaptive response to alkylation stress. And this response is regulated by a DNA methyltransferase called ADA. And this ADA protein senses DNA methylation lesions, such as those generated by the MMS drug that I talked about earlier. So this ADA protein removes those methyl groups from the DNA and transfers them onto a cysteine residue on itself. And this methylated ADA protein then turns into a transcription activator of its own gene, producing more ADA molecules. These ADA molecules can then further search for DNA damage, become methylated. So overall, you get this positive feedback loop that increases the amounts of ADA proteins per cell. So when we treated cells that express the fluorescent fusion of ADA to the fluorescent protein MYPET with the DNA methylating agent MMS, after 1 hour, we found that many cells strongly induced the expression of ADA, but there were also several cells that didn't seem to respond at all and still had the background fluorescent levels that you see in untreated cells. And even with high concentration of MMS, you see that there are about 20% of cells that seem to fail to induce this response. And that was surprising, given that DNA alkylation damage is toxic and mutagenic to cells. You'd expect that these cells would want to very accurately and rapidly induce this DNA damage response to survive and maintain their genome stability. So we looked at these cells over time using a single cell imaging technique. So this involves microfluidic channels into which individual E. coli cells are inserted. And we can watch the growth and division of these individual cells over time. And we can simultaneously flush in growth medium and DNA damaging treatments. And so when I play the movie, you can see that initially the cells only express the cell marker, which is a fluorescent protein that we use to automatically segment and identify the positions of the cells. And then in yellow is the expression of the ADA fluorescent fusion. So you can see the cells are growing and dividing in these channels. The progeny at the top of each channel is being flushed out and washed away. And that allows us to watch these individual lineages over very, very long times under very constant growth conditions and constant treatment conditions as well. So what you see here is that many cells very quickly activate the ADA response. They turn bright yellow. But now many generations have gone by, and there are still some lineages that have yet to sense the danger. They still haven't induced the ADA response. They still keep on growing as if there is no problem. And this was obviously surprising. We wondered why there's such variation between these individual cells that are growing under very much identical conditions, and they all derived from a single colony of genetically identical cells. So the question, of course, was, does this heterogeneity that we see between these individual cells? Does it have a purpose, or is it just noise? Is there some benefit of this heterogeneity for the function of these cells, or is it just that the regulation of this DNA damage response is inherently noisy and the cells can't help it, but some cells activate the response well and other cells fail to activate it? So to address that question, we looked at several different aspects. We asked whether this response, the way it's genetically wired, is it wired to create variation, or is it rather than suppressing it, is it trying to be accurate, or is it actually trying to be noisy? Does this variation create some distinct phenotypic states that have defined properties? Could this variation actually be avoided, or is this an inevitable part of this response? And does this variation potentially have a functional benefit for cells? And ultimately, maybe is this conserved between different species? So initially, we looked at the genetic network that underlines the ADA response. So I mentioned before that this is a positive feedback loop where the ADA protein is generated from the ADA gene, becomes methylated, and then induces its own gene. Now, what we found when we looked at the amount of ADA proteins in undamaged cells, we found that it was extremely low. Here we used single molecule imaging and simply counted the number of spots that we get of ADA proteins per cell. And this was very, very low. In fact, 20 to 30% of cells had not even a single copy of ADA. Many cells had 1 or 2, maybe sometimes 3 copies, but the overall mean expression was just 1 molecule per cell, and many cells had 0 copies. Now, what does that mean for the regulation of the response? Well, we simulated the same distribution and simply simulated a constant rate of ADA protein production in the absence of DNA damage with a mean rate of 1. But of course, if the mean rate is 1, sometimes you might generate 1 molecule per cell generation, sometimes maybe 2, but also sometimes 0. So this is a stochastic process that leads to this observed Poisson distribution of protein copy numbers per cell. And just with that as an input into our simulation, coupled to this positive feedback regulation, we very closely recapitulate the time traces of fluorescent protein production in individual cells, where some cells activate the response very quickly, whereas other cells might wait for 10 or even 20 generations before they induce this response. However, once the DNA damaging agent is removed, then all the cells deactivate the response simultaneously together. It just shows that the cells are actually able to sense the DNA damage level very accurately and are able to respond accurately. And it turns out that the heterogeneity in these activation delays can actually be avoided. So when we have wild-type cells and we treat them with MMS, we see this mixture of non-responding and responding cells. But if we inhibit cell division before treatment, then fewer cells are in that state with 0 molecules of ADA because they haven't lost individual molecules at the process of during cell division. And so in these filamentous cells, we now see a uniform induction of the ADA response. Furthermore, if we slightly overexpress ADA expression in cells, then this leads to a very uniform response. And we also found a point mutant where one of the cysteine residues that's involved in sensing DNA methylation damage is mutated to alanine. Then we see a more uniform response where cells even activate the response before DNA damage treatment. Now we asked if this variation in ADA levels actually affects the whole regulon of this response. We found that it actually generates 2 very distinct subpopulations of cells. So besides ADA, there's another DNA repair enzyme called ALK-B and another one called ALK-A that are also controlled by ADA. We found that both ALK-B and ALK-A are also heterogeneously activated with long delays in the activation time for some cells. And even though this looks very noisy, it turns out that these genes all activate simultaneously at the same time. So here's a dual reporter where you looked at the fluorescent protein expression for ADA and ALK-A at the same time using 2 different colors. And you can see that there's a stochastic delay before these proteins are activated, but once they're activated, they both activated exactly at the same time. So here you can see the distribution of the activation times for ADA and for ALK-A, and each dot here is a single cell showing the spread of activation times across cells, but they fall on a line showing that they activate at the same time. And furthermore, the expression levels of ALK-B and ALK-A before any DNA damage treatment are extremely low. So this shows that we really have two distinct populations of cells. There's a subpopulation of cells that fails to activate the response. They essentially have no copies of these DNA repair enzymes. And then there are the cells that activate, and they activate these different DNA repair enzymes at the same time to very high levels. It turns out that this delayed activation of the ADA response has a temporary fitness cost. So if we have mutants of these enzymes that can never induce this response, they're very sensitive to the treatment with methylation damage. As you can see here, very few colonies form on a plate with MMS treatment. We look at the survival of these cells over time. You can see in untreated cells in the fluidics channels, there's very good survival as the cells are growing in the channels. Whereas when we treat these cells with MMS, we see that the survival drops, and we can now split the populations between the cells that have activated the response in green and the cells that have failed to activate the response in orange. And initially, the cells that have failed to activate the response, they decay at the same rate as the mutant strain, that delta ADA mutant strain that lacks this response completely. But over time, the ADA off cells also activate the response and eventually have a better chance of survival compared to the mutant cells, which can never activate the response. And that means that this delayed ADA activation only has a temporary fitness cost for the cells. So how does this variation in DNA repair affect genome stability? So DNA methylation damage is toxic, but it is also very mutagenic. So how does this variation in the ability to repair such mutagenic DNA lesions actually affect the stability of the genetic information? And this is a question that's difficult to address with conventional methods, because even though you can sequence cells, you can find out which mutations have happened, but this is the final end product of mutagenesis. You can see the mutation, but you cannot really see which molecular processes cause the mutations. You cannot distinguish between different subpopulations of cells. So to address this question, we use a fluorescent imaging-based technique to directly look at the dynamics of mutagenesis in individual living cells. And this is based on using a reporter system that uses a fluorescently labeled DNA mismatch repair enzyme called MUT-L. And this MUT-L-MY fusion binds to DNA mismatches, which are the initial site of a DNA replication error when the base is mispaired with the wrong base pair. And these MUT-L-MY fusion, they form foci at these sites of DNA replication errors. So here you can see in individual living cells, we can detect these bright spots that correspond to nascent sites of DNA replication errors. So we can then look at the frequency of these DNA replication errors and compare the frequency of this to the timing of the DNA damage response in the same cells. So here's the fluorescent level of the ADA response that I've introduced before. And then in the same cell, we can look at the frequency of DNA mismatches. We find that these DNA mismatches fall into a pulse, and this pulse with a much higher frequency of DNA mismatches. The timing of that matches very closely the timing of the delay of the ADA response. So here we've got unadapted cells initially that then suddenly experience DNA damaging treatment because they have not yet induced the protective DNA damage response. They have a higher frequency of DNA mismatches. And then at some point, each individual cell switches on the ADA response, which increases its capacity to remove the mutagenic lesions. And that allows the DNA replication error rate to go back down again to the baseline level. And indeed, when we have a mutant, delta ADA mutant, that cannot induce the protective DNA damage response, then a high rate of DNA replication errors is maintained and the cells do not adapt. And then the nice thing with this is that we can actually do this at a single cell level. So here at the top, you see one example time trace of one cell that activated the ADA response quite early, and it had one DNA replication error in this fairly short delay of just about one generation time. And here below, you see a late responding cell that waited until 3 or 4 generations of treatment before it activated the response. And this cell had a longer period of increased replication error. So overall, we see that the cells try to balance DNA repair and mutagenesis during DNA alkylation stress. And during this alkylation stress, initially, the cells experience a period of increased mutagenesis. And then they eventually activate the ADA response, which increases the capacity of the DNA repair system. But this is associated with intracellular noise. Some cells activate this very early, and some cells wait for much longer time. So this kind of flips some ideas about the sort of central dogma in genetics on its head. So normally we think about genetic heterogeneity being the source of phenotypic heterogeneity. Whereas here, we've got initially genetically identical cells, but these cells have phenotypic noise, they have variation in their ability to sense DNA damage, and this leads to differences in mutation rate, and it could be a source of genetic heterogeneity. And in fact, maybe such differences in mutation rate could be a useful property for cells in a population. So if you imagine you initially have a population of E. coli cells, they suddenly experience adverse conditions in their environment. And so what can they do? Where they might need to mutate in order to adapt to these new stressful conditions and become resistant. So it could then be beneficial if a subpopulation of cells switches to a state where they have higher mutation rates while the rest of the population maintains low mutation rates. Because if the stress persists, there's a chance that these cells with higher mutation rates acquire a beneficial mutation, which makes these cells then resistant to the stress. And all the other cells might die, but this one of these mutator cells that had a higher mutation rate and gets a beneficial mutation could then enable population survival. This is a process called mutational rescue. On the other hand, if the stress passes, and the cells at the time of stress cannot really predict whether the stress is going to stay or pass. If the stress passes, well, then it's good that the majority of the population didn't switch to a state with higher mutation rates. They were able to maintain the genome stability, the optimal evolved genotype that was optimal for the environmental conditions that this organism has been in. And so over time, the subpopulation of mutator bacteria switches back to its normal state, and overall the genetic stability was maintained. So we then ask, does such mutation rate variation possibly benefit population evolvability? And to do this, we used a cell sorting technique where we first induced the ADA response, which leads to that stochastic variation between cells that are in the off state, which have higher mutation rates, and cells in the on state, which we think have lower mutation rates. And so to test this, we then sorted these cells with a flow cytometer and then plated these on the two populations separately on LD plates containing rifampicin antibiotic. And so these cells are able to grow on a plate containing rifampicin. It means that they acquired a mutation in the RNA polymerase gene, the gene that leads to rifampicin resistance.
Do you have, sorry, about 3 minutes? It's okay? Or
yeah, it's okay. I can.
Just to kind of, we have some time for the questions. I'm just thinking flow cytometry experiments take a long time sometimes.
I wrap up now. Good.
If that's okay, thank you.
Perfect. So what we can then see is that after treatment with MMS, we see that the on population had a low frequency of rifampicin-resistant colonies, whereas the population with a higher mutation rate, that was in the off state, had a higher mutation rate. We see that also for increased amounts of MMS treatment, whereas at even higher levels, at 10 millimolar MMS, we see that there's now also a contribution of cell killing, where also the overall number of colonies goes down. But in each case, we see that the off population had a higher mutation frequency compared to the on population. Right. So I'm going to fast forward a little bit in the interest of time, because it'd be nice to talk about, have some time for questions. So initially, I've been talking about single molecule imaging techniques to look directly at the DNA target search of DNA repair enzymes, as well as enzymes involved in transcription and replication, etc. And we find that these enzymes search for their target sites by a combination of free diffusion and non-specific binding to DNA. And we surprisingly found that the contribution of non-specific binding to DNA vastly outweighs the contribution of free diffusion. We also looked at how cell-to-cell heterogeneity in DNA repair could balance or perturb the balance between DNA repair and mutagenesis, and this could have some interesting effects on the dynamics of evolution of bacterial populations to changes in the environment. And overall, we're interested in this interplay between how both phenotypic changes and genetic changes contribute to evolution of bacteria. And I think our microscopy techniques are pretty powerful in being able to both resolve phenotypic dynamics in bacteria, as well as also look at the dynamics of the mutation supply that drives genetic changes. And so let me just thank the people in my group that have contributed to this work over the years, funding agencies, excellent collaborators and mentors over the years. And thank you very much for your attention, and I'm looking forward to addressing your questions. Thank you.
Great.
Thank you very much. I think the adventure is to begin now. So first, thank you very much for the talk. And Nick, if you could engage the Mentimeter.
Right. Stephan, if you could unshare, I'll bring the quiz up.
Perfect.
And I think maybe within the interest of time, you can also, I guess the quiz has something related to the question. Maybe we can, probably take on that. Also a timely challenge. Thank you very much, Stephan.
So the link for the quiz is in the chat. You should see that. Okay, we've got people joining. Great prize again this week, the fold scope. So fingers on the buzzers for that. Okay, let's see if we can get a few more people joining us. Well, we got 6 people. We're getting there. Make it worthwhile. Yeah, that was extremely interesting. I've got a few questions whenever we get to those, but let's see if we can get another person joining us. One more person, come on. I know you're out there. The fold scope is a fine prize. All right.
I think we need to kind of up maybe our prizes.
How could you improve on that? I don't know.
It's easy to ship.
All right, okay, we got another person. Right, I think I will launch the quiz. So with that, let's go into question number 1. All right, everybody in? Yep. And here we go. So the first question is, how do many DNA binding proteins locate to their target sites on DNA? Is that by 3D random diffusion, by using energy from ATP hydrolysis, or by non-specific binding and sliding along the DNA? And here we go. Let's see who's got the fastest response. Okay, got some good answers there.
It's fast.
All right, let's see what the scores are so far. And Barefoot Contessa, I think, is in the lead, but there's plenty of people on your tail. So if you're quick, you can get the prize. Let's go on to the next question. Question 2. Photoswitchable or blinking dyes are useful for single molecule tracking of proteins in cells because of a large number of molecules per cell can be tracked sequentially. There's less concern that the dye perturbs the function of the protein, or the dyes are generally very bright and less prone to photo bleaching. And the answer to this question is a split opinion there. Okay, let's see what we've got in terms of the leaderboard now.
Good work, people.
Yep. Come on. Right, here we go. Let's see if anything changes. CJ has now.
CJ, you win.
Into the lead. Well done. Okay, next question. At the single molecule level, photobleaching leads to a gradual fading of fluorescence emission from a molecule, or the sudden disappearance of fluorescence emission from a molecule, or photobleaching does not affect single molecules. Let's see what everyone thinks of this one. Yep, so we've got sudden disappearance. Okay, so let's see what the leaderboard looks like now. Ooh, things might have changed. Yeah, I think we've leaders, places have changed. Tuito and Alex neck and neck.
Oh, this is thrilling.
Let's see how this goes in the next question. Question 4. 2 more questions. E. coli shells show variable expression of the adaptive response after DNA damage treatment because the basal number of ADA molecules is very low. Mutation rates differ between individual cells, or the amount of DNA damage is variable across cells. Let's see what the answer for this one is. Let's see what people think. Right.
Sorry, but
There we are. And the basal number of ADA molecules is very low is the correct answer for that one. Let's see if I can get the leaderboard up again. Oh, wow. Another change in leadership, perhaps. Yeah, CJ back on top again. Okay. Right, well, CJ, Tweeto, Alex, and Pick, I think.
What's down to the last question?
It's all about speed now. Any one of you could win. Let's see. And here we go, last question. MUT-L is a, or MUT-L is a DNA nucleotide excision repair enzyme, a DNA mismatch repair enzyme, or a DNA methyltransferase enzyme. I definitely heard the answer to this one in the talk. It's a DNA mismatch repair enzyme. So let's see who clinches it in the end. Oh, Alex.
Wow.
Alex. That was an interesting quiz. I mean, I love the questions, but also
very nice, yeah.
It was changing leader throughout. So Alex, if you could put your details into the chat, then someone from, oh, well done, Alex.
Yeah.
Well done, Alex.
And thank you very much for the questions, Stephan. That was quite good fun there. Yeah,
it was good fun. Yeah.
Okay,
Nice.
so please could people put their questions in the chat or stick their hand up to ask them. So we got Cynthia, Becky, Sue, please see. If you'd like to unmute, you could answer your question in person. Otherwise, I'll just read it out for you. Are you there? No? And while we're doing that, other people, if you've got questions, please put them in there. Yes, but no mic. Okay, I'll read it out. Sorry. What is the definition of apparent diffusion coefficient? Sorry about that, Cynthia.
Yeah, no, that's a good question. Now the quiz questions come back to me, because that's also a good quiz question. But so we, first of all, use apparent diffusion coefficient often as a way of saying that this is not the true sort of correct microscopic diffusion coefficient, in the sense that our observation of the diffusion coefficient has some biases in it that have to do with the way we measure it. So when we measure it, we find the locations of a protein inside the cell and link them together, and then we calculate the mean squared displacement from these. And Brownian motion theory says that the mean squared displacement increases linearly with time. And so by basically looking at the slope of how the mean squared displacement increases with time, we can get the diffusion coefficient, which is the slope of that curve. Now the problem is bacteria are very small, and even something that moves very fast can never get outside the volume of the cell. And so that means that we always underestimate the actual true diffusion coefficients, because the confinement of the tracks within the cell volume mean that we always measure smaller distances than a molecule would actually move if it was arbitrarily in a very, very large volume. And so that's what we call it apparent diffusion coefficient. Another reason is that the true positions of the molecule are perturbed by the localization error for each localization. So because of our optical imaging technique, we cannot say with arbitrary precision where an individual molecule is. And so that little localization error is basically adds a small random step to every genuine localization that the molecule has. And that, on the other hand, leads to a slight overestimation of the real diffusion coefficient. And we can correct for these effects by taking into account the cell geometry, and by measuring the localization error, subtracting that contribution. And so that's when we measure basically accurate diffusion coefficient. But for many of our measurements, we, for instance, just want to distinguish between mobile and immobile populations of molecules, we don't care exactly what the diffusion coefficient is, as long as we can tell different populations apart. So for those plots, we like to stick with the raw data, which we call apparent diffusion coefficient. Sorry, that was a long answer, but hopefully addressed your question.
Well, that's an important point. So I've got a question from Charles Winterholt. Do you want to unmute and ask a question, Charles?
Yeah, sure. Can you hear me now?
Hello, Charles.
Yeah, hi. So in Bacillus, the AD and LKB genes are encoded in prophage elements. And I was wondering whether that contributes, well, whether that's the case in E. coli, and whether that could contribute to the response you observe in there.
Yeah, it's not the case in E. coli, but it's very interesting. Yeah, in different bacterial species, the ADA response genes actually very much conserved. So you find these proteins in various different bacterial species, but the regulation is often different. In some cases, the different domains of the proteins are present in different peptides, so it'd be quite interesting to see, and something that we're now working on, to see if this heterogeneity is a conserved feature, even if some of the details of the regulation might differ between species. So if we find that this heterogeneity is something that we still see in different species, that would also really indicate that this is some beneficial feature, and that's been conserved. So I think it's very interesting. I'm not sure about the regulation of the prophage gene in B. subtilis, but yeah, often phage repressors are also very heterogeneously expressed and triggered. Prophage activation is often triggered by DNA damage, so that kind of interplay will be very interesting to explore.
Brilliant, thank you very much.
Mm-hmm.
All right. Do we have any other questions in there? Oh, well, people are thinking of some other questions. Actually, I do have one. So, have single molecule fluorescence tracks got longer due to improvements in technology? Is this something you've seen?
Yes, for sure. So basically, as our instrumentation and dyes are becoming better, we can basically get away with using lower excitation intensities, which reduces photo-bleaching, which means we can see individual molecules for longer. One of the biggest changes that we've capitalized on was that we've more or less moved away from using photoactivatable fluorescent proteins and instead use genetically encoded protein tags, such as the halo tag, which can then be covalently labeled with bright synthetic fluorophores, which are able to penetrate into live E. coli cells. And so we can use dyes like TMR, which are much brighter than photoactivatable M-cherry, for instance, but have quite similar spectral characteristics. And then conveniently, TMR, for instance, also blinks inside live cells. So it's a bit different. So initially, you've got all the molecules in a fluorescent state, and then over time, with illumination, the molecules become non-fluorescent and switch to a dark state, but then become reactivated. And so when they're in the bright state, they stay in that bright state for quite a bit longer than the photoactivatable M-cherry does. So you can more easily track these molecules over long times.
Great, thank you very much. Do we have any other questions from the audience? Go on, if anyone puts their hand up.
I think there was another one from Cynthia in the chat.
Oh yeah, can you? Yeah.
What about the link? So what's the link algorithm used by the measurements? So this has to do with the single molecule tracking technique. So we're using an algorithm that simply looks at the nearest neighbor from one frame to the next. So imagine you've got your distribution of spots that have been localized in one frame, and then the next frame, another distribution of spots that has been localized by our spot finding algorithm. And that list is then simply compared, and the localizations are linked together if they appear within a user-defined window. And if they're more than 2 spots within such a window, then the sum of the distances is minimized, such that it's most likely that the two spots that belong to the same molecule are linked together. Of course, diffusion is random, and such an algorithm will inevitably make mistakes. So we try to keep the density of the molecules that are fluorescent at any one time as low as possible, such that the majority of spots only have one possible link from one frame to the next. There's a memory parameter. So if a molecule sometimes disappears for a frame or 2 and then reappears, such as happens due to photolinking, then we can still link those molecules together. Okay. And how do we define? Sorry, yeah.
Carry on. I can see you're reading them faster than I am getting them, so carry on.
Yeah, sure. Cynthia, you're then asking how we define the size of the window. So this has to do with the diffusion coefficient that we expect for molecules. So we basically we start with a sensible choice of window, and then we look at the distribution of step sizes. And if our window is too small, then that distribution of step sizes kind of is cut off, where we're not allowing any steps that are larger than that window. And then we gradually increase the size of the window, such that we basically get a smooth distribution that seems to capture the majority of the steps.
So actually, I have maybe one more question, which if no one else is chipping in, I'll be getting some thanks there. But how does your work speak to the problem of antibiotic resistance, or the general health problem that we all have to face in the coming years?
Yeah, thanks, Nick. So we're actively working on this, having characterized our imaging techniques using kind of defined DNA damage treatments like alkylation damage and oxidative stress treatments. We've also begun to work with the stress that are caused by different types of antibiotics. And some of these antibiotics directly generate DNA damage. So you see an increase in the replication error rate and mutation frequency because of that antibiotic treatment. And for some antibiotics, it's not as obvious because they target the cell wall, or they target protein translation. Yet the stresses that are caused by these antibiotics seem to also increase the production of reactive oxygen species, which can cause DNA damage and thereby increase frequency of DNA replication errors. So that's something we're quite interested in, how the regulation of the stress responses to the antibiotics and the dynamics of mutagenesis play together, and ultimately can lead to either cell killing or evolution of resistance.
Right, thank you very much. So I think we've reached about time, so I would just say, get everybody to join me in thanking you for such a fantastic talk. That was really interesting, and I love single molecule fluorescence measurements. They are just really amazing things to be able to do, and very powerful in your hands, obviously. So great, thank you very much for that.
Thank you very much, Nick, and thanks everybody for questions and joining us today. And I hope I get to meet some of you in person at some point. Stop me if you see me at a conference or so.
There you are. There's an invitation from an expert. So thank you very much, Stephan. And with that, I'll just give you my own round of applause and then bid everybody goodbye.